Hello students, today we are going to start a new chapter that is Golgi bodies. As this is a part of cell, the cell organelle which we are going to study is Golgi bodies. Golgi bodies was discovered by Camillo Golgi and it is also known as Golgi apparatus. And it is also known as lipochondria or iodosomes. As it was discovered by Mr. George in 1867, now, Camillo Golgi in 1898 he isolated that thing from the nerve cell of the fat, where basically it is found. Golgi body is absent from the prokaryote. It is present in only the eukaryote except mature mammalian cell. Now, if we see how the Golgi ultrastructure of Golgi body look like, this is how it look like. Okay, so this is you can see very transition vesicles, sacles, cisternae membrane, and vacuole. So basically it is also known as the warehouse of the cell where the food stored synthesized is stored over here. So it's basically structure of Golgi apparatus consists of cisternae, flattened sac-like structure, vacuoles, they are spherical, component lies towards the concave side of the cisternae, vesicles. So this is how it look like. Now what are the functions of Golgi complex? First function is it involved in cell secretion. So its large size is secretory cells. Second, what it is? It acts as a packaging membrane. Okay. Third is it can be parts in cell plate formation. For the formation of cell plate, it takes plays an important role. Next is synthesis of peptic substances. The peptic substances are being synthesized into it. Next is it helps in the formation of acrosomes of sperm. Okay. Next, it is also helps in the formation of primary lysosomes, and it helps in the biosynthesis of glycoprotein and glycolipids and glycosylation, and it also helps in the recycling of plasma membrane. So these all are the functions of Golgi complex. Now let's move on to the next one, that is ribosomes. As you all know, ribosomes are responsible for the synthesis of protein. It's very important function. So ribosome was first isolated from the cells of cytoplasm by Claude in the year 1943 and were named as microsomes. Okay, these were reported in plant cell by Robinson and Brown in 1953. These are called ribosomes by G. Pallid. So the term ribosome was given by G. Pallid in the year 1955. Now, ribosome basically formed of two set of pair of structures. So on the basis of that, ribosomes are classified into two groups, that is 70S and 80S ribosomes. So now you can see the first type of ribosome, it is of 70S and it is a prokaryotic type of ribosomes. It is prokaryotic in nature. In this you can see we have 30S and 50S joined together to make 70S ribosomes. Its length is 200 to 290 Armstrong and its width is 170 to 210 Armstrong width होता है. Now coming to the that is eukaryotic ribosomes. In case of eukaryotic ribosomes, the upper portion is of 40S and lower is of 60S. Combining it is forming 100, but it is 80S because 20S size has been reduced just because of the submersion of both the part. So what will be the length of this ribosome? It is 300 to 340 Armstrong and width will be 200 to 240 Armstrong. So this is the size of the ribosome, which are basically found in two types of basically ribosome is of two types, that is 70S and 80S. 70S is found in prokaryotes and 80S is found in eukaryotic cells. As a ribosome performs various functions and they used to get attached with the RNA and forms a polysome or polyribosomes, means attaching, it simply means a number of ribosomes attached in a sequence from the structure. So the structure of ribosome, I think it is very much clear to you all that it is of two type. It can be of two type, that is 70S and 80S. The function of ribosome is very much clear as it is considered as a synthesis of protein is being done over here only. So complete synthesis of protein is be done over here, and ribosomes are basically responsible for this. Okay, so basically if we conclude about 70s and 80s ribosomes, so there is a major difference between it, like sedimentation coefficient, that is 70s and 80s, means we can say on the basis of sedimentation how they are settled, they are classified into two groups, that is 70s and 80s. Size, as you all know, one is 70s is of 200 to 290 Armstrong, while 80s is of 300 to 340 Armstrong. Molecular weight of prokaryotic ribosome is 2.8 into 10 to the power 6 Dalton, while eukaryotic size is molecular weight is 4.5 to 5 into 10 to the power 6 Daltons. Number of RNAs, how much prokaryotes hold, that is 3 types, and eukaryotic 4 types. Okay, now larger subunits. Sedimentation coefficient, we will see the largest subunits in prokaryotic is 70s, while in eukaryotic it is 60s. Molecular weight is also different. Prokaryotic is having 1.8 into 10 to the power 6, while 60s is having 3.3 to 3.5 10 to the power 6 Armstrong. Okay, now types, 2 types prokaryotic and 3 types, that is of eukaryotic, while protein, the prokaryotics synthesize 34 type excuse me, while eukaryotic synthesize 40 type. Same with the smaller units, one is having 30 as a smaller unit, and eukaryotic is having 40. So it is very much clear with this figure. Okay, now coming to its function. So what is the function of ribosome? The function of ribosome, first function, it is given, ribosome are called protein factories or engines of the cell, because these are the site of protein synthesis. So protein controls either cellular structure and or act as a enzyme and controls the cellular functions. Basically, whatever the place ribosomes are there, they are responsible for the synthesis of protein, and that's why they are considered as engines. They basically act as enzymes for the synthesis of protein. Now, on the next function is the free ribosomes produce non-secretory protein is enzymes of intercellular use, means with the ribosomes which are free, they basically produces non-secretory proteins which are used in the as a intercellular use. They are basically used in the intercellular purposes. Last is bound ribosomes or RER synthesis secretory proteins, enzymes for the extracellular use. Example, pancreatic cells, liver cells, plasma cells. So what is meant by the bound ribosomes? Ribosomes which are attached to the surface are considered as a bound ribosomes. So these are the two cell organelles which we have discussed today, that is which one we have discussed, the Golgi complex and ribosomes. Golgi complex are basically originated from the endoplasmic reticulum. The scientist associated is Camilo Golgi, while ribosomes are responsible for the protein synthesis. The scientist use it is Mr. G. Pallid, and it has various functions. So students, this is all for the day. Thank you.